Okay, sorry about that. So first of all, it's a pleasure to be here. It's always a pleasure to be here. I'm honored to be a part of this group. There's some amazing scientists that are working on this, and it's very humbling and very appreciative of this opportunity that the FAST is given me to be involved in this. So I was asked to speak for 20 minutes, and as those of you who know me, that's going to be very difficult, but I'll do my best here. I'm going to try to keep track of my time. What I thought I would do was, if we can, is give you just a brief overview of what we're working on as part of the FIRE project. And what I'm going to do is give a little bit of background in terms of why we're doing the things that we're doing. A little bit of data. A lot of this you'll just have to take my word for it. I'll try not to go into the science too much. And then maybe a little bit about where we hope to go with these experiments and the things that we're doing. Really what we're trying to do here is develop a therapeutic. I mean, along the way, we are interested in learning a lot about UB3A and how the gene is regulated. But as you know, this team was put together to develop therapeutics and, as you can understand, to develop them as quickly as possible. So these are the three areas that I'm going to briefly talk about. So one is our interest in understanding the mechanisms that regulate imprinting of UBE3A in neurons. And so this is a very unusual gene in terms of the way that it's regulated in the brain. And so I've been interested, and my lab's been interested in understanding how this process occurs and also why it occurs. And that's more of a basic science question, but understanding the how and the why will help us develop new strategies to circumvent this process and to understand what we're doing if we do in fact do that. We're also actively involved in the identification of new treatments or therapies for AS, and what we've been doing is developing high throughput screening assays so that we can look at as many drugs and compounds as quickly as we can. It takes a lot of time, a lot of money to do this, and so we've been actively trying to develop new strategies to expedite this process. And then last, I'll mention our work to develop new models of Angelman syndrome. It's been mentioned there's a mouse model of Angelman syndrome that's been extensively used. We're in the process of developing a large animal model of Angelman syndrome, and I'll sort of go over why we think that's important and so forth. Before I get started, I just want to point out the people that are in my lab. Some of them have left. Most of them are still here. We've hosted some students and so forth from other places, but this is really the team that I have. Much of the work that I'll talk about today was done by Dr. Paul Hillman, Dr. Sarah Christian. She was a postdoctoral fellowship through FAST, and then Jade Benjamin, who's currently a graduate student in my lab, as well as Hillary Shaheen. So why we want to understand how UB3A is imprinted, or the mechanism by which UB3A is imprinted in the brain, is so that we can figure out ways to circumvent this process. So as you've heard, one of the approaches that's being used is to develop strategies that reactivate or activate expression of the paternal allele. And I'll talk about that in my second part of this talk. But if we understand how the paternal allele is turned off in neurons, the idea is that we may be able to develop a way to turn it back on. And it's really just as simple as that. I mean, there's a lot of work that has to be done in this area to know whether or not this is an effective treatment. But the reality is that all of us in this room, as far as we know, have our fathers, the allele that we inherited from our father of UB3A, has been turned off. Why that happened, nobody really knows. But nonetheless, that allele is off. It seems to be the natural state. And all we want to do is try to turn it back on as a therapeutic. And the idea is that the expression of that allele will replace what's been lost through a mutation or deletion on the maternal chromosome. And so, it's going to be a problem here. So as you've probably seen in terms of gene regulation, this region in the genome of chromosome 15q11, q13 is perhaps the most complicated region in terms of its allelic expression and the epigenetic factors that regulates expression. There is, without going into a lot of details, what we know about this locus is that on the maternal chromosome, and this is in the central nervous system, UB3A is expressed, whereas all these other genes are not. On the paternal chromosome, these remaining genes are expressed, and UB3A is turned off. The way that UB3A is turned off is through the expression of this long non-coding transcript that involves a number of regulatory RNAs. And so we think that an activation of the paternal allele is actually linked to the expression of these other regulatory RNAs, almost like they're competing with each other in such a way. But the idea is to understand precisely how this long non-coding antisense transcript turns off expression of the paternal allele. And so there's a number of models that exist. We know that the expression of this long non-coding RNA, it's only expressed in neurons, so in all the cells of our body except neurons of the central nervous system. UB3A is expressed from the paternal chromosome, but in neurons, this long non-coding transcript is expressed, it's highly expressed, and by some unknown mechanism, it inhibits transcription of the paternal allele. And it's a very unique mechanism because most imprinted genes that are regulated by an antisense transcript involve repressive histone modifications or these epigenetic modifications that silence transcription of the allele. That's not the case for UB3A. For UB3A, the paternal allele is actively transcribed. It just doesn't complete the process of transcription. So somehow the antisense transcript is stopping the paternal allele from completing a full-length transcript. And so I won't go into any details here, but what we've been trying to do is to understand why this happens. You know, what's the biological relevance of turning off the paternal allele, and then how this happens. And we've focused on one particular area, and I'll sort of quickly go through this. This is going to take forever with this. How about when I hold that up, y'all? Can y'all move it back there? There we go. And so what we've done is we've looked at both the mouse and human UB3A gene, trying to understand why UB3A is imprinted. We find that it doesn't appear to be a dosage regulating mechanism, so the inactivation of the paternal allele doesn't appear to have evolved as a means to reduce the amount of UB3A in the brain. In fact, we see that UB3A levels are higher in neurons than they are most everywhere in the body. We've done sort of evolutionary studies that have shown this to be the case. We've looked at the establishment of the imprint during neurogenesis, and what we see is that the expression levels of UB3A are tightly regulated, even though one allele is being turned off. And so for whatever reason that is, that it's being turned off, some mechanism within the cell, within neurons, is maintaining the level of UB3A, despite the fact that one allele is being turned off. And that's more related to the why than the how. Regarding the how, we've been focusing our attention on the antisense transcript and trying to understand its expression levels, how it's been processed, where it goes, where it stops, and been looking at this in both in human and mouse and trying to understand exactly how that long non-coding RNA transcript turns off the expression of the paternal allele. And we see that it's very unique. We have a model that we've put together, and this is something that we're actively working on in our lab. Much of this, though, it's very basic in nature, but the idea, again, is that if we understand how the process occurs, then we can somehow perhaps circumvent it. Now we're going to have to get this fixed. So the next part is the identification of new therapies for Angelman syndrome, and this is very much related to the first part of my talk. And so there are a number of efforts that are going on, not just with the FIRE team, but globally, where people are looking for new therapies for AS. And the approach that we took was to speed this process up. And there are a number of groups, pharmaceutical companies, what have you, that perform high throughput screening assays to identify potential candidates for new therapies. And so what we wanted to do is take the knowledge that we had and some of the tools that we had and use that and see if we could develop new ways to rapidly screen drugs as potential therapies for Angelman syndrome. And so I want to point out some of our collaborations that we have at Texas A&M. This particular project it relies heavily on the Texas A&M Institute for Genomic Medicine. Drs. Ben Merpergo, Andre Golovko, and Dr. Jonathan Ballard have been instrumental in helping us set up this particular part of this project. And you'll kind of see how this unfolds here in a second. In addition to that, we work closely with the Texas A&M Institute for Biotechnology that's located in Houston, Texas. And that facility there is a high throughput screening facility that has hundreds of thousands of potential drugs, small molecules and FDA approved drugs that are available to us at essentially no cost. And so we've been working with these two groups to develop a new strategy to screen these libraries for drugs that can reactivate expression of the maternal allele or that can be used as a treatment for the symptoms. And so there's two approaches that we've been taking. The first is a target-based approach, and the second is a symptom-based approach. And so in a target-based approach, we're looking for a therapy that targets UB3A. And so in these particular assays, they're biased. You know, what we're trying to do is reactivate expression of the maternal allele. Our outcome measure is only one thing, and that's expression of the maternal allele. And it's very focused. The problem with these types of studies is that they are biased. And so if we're only looking at one thing and nothing else and we don't see anything, well, you know, there are things that we might have missed. The symptom-based approach is an unbiased method. And here what we're attempting to do is to look for compounds or drugs that mitigate the symptoms that are associated with AS. And so in this particular strategy, we're not particularly interested in the mechanism by which this occurs. We simply want to see if something works. This is thought to be a much more powerful approach because, quite frankly, we're not trying to outsmart the biology that's associated with this condition. We throw the kitchen sink, if you will, at the condition and we see what makes it better. We really don't care what it's doing. We're simply looking to see if the symptoms improve. And so we've taken these two strategies and tried to develop high throughput assays to find a new therapy for AS. So in the first target-based approach, what we're doing is looking for strategies that will reactivate expression of the paternal allele. And so as I mentioned, we know that the expression of this antisense transcript or this long non-coding RNA is necessary and sufficient for inhibiting expression of the paternal allele. And so the approach is quite simple. We're simply looking for things that will inhibit or interfere with this process with the idea that it will reactivate expression of that paternal allele. And this approach has been used. I'm sure we've already mentioned it. There, in some of the earlier talks, and I know that some of y'all are probably familiar with this, the work that Mark Zilka did and Ben Philpott did at UNC, where the topoisomerase inhibitors were found, are shown to be able to reactivate expression of paternal allele, as well as Dr. Baudet's group at Baylor College of Medicine, where they found that these antisense oligonucleotides that targeted the antisense transcript were also capable of reactivating expression of paternal allele. And so we didn't want to reinvent the wheel or redo what they did. We actually wanted to make this more powerful. And so with these types of studies, especially since UB3A is only imprinted in neurons of the central nervous system, they're complicated because you have to have a primary cell line that you isolate from the mouse brain. So neurons are post-mitotic, they don't grow in culture. And so each time one of these experiments are done, we have to sacrifice a mouse, we have to extract its brain, we have to isolate neurons from that mouse, plate it in a dish, and then add the compounds to it. There's only a limited number of neurons in the brain that you can culture. So when we typically use the hippocampus, there's roughly 1.5 million neurons that you can extract from one mouse hippocampus. That gives you, you can translate that into a certain number of compounds that you can screen, which is not that many. So the conventional way of doing this is very labor intensive, largely because of the source of the cells that are used to screen the drugs. So what we decided to do was to use a different approach where we isolated embryonic stem cells from mice that had a YFP reporter allele on the paternal chromosome. And so these mice were developed while I was at Baylor College of Medicine, and it's basically a knock-in mouse model where the gene for the yellow fluorescent protein was targeted to the 3' end or the end of UB3A. And what's nice about these mice is that you can use that fluorescent tag to look at the expression of either the maternal or paternal allele. And so these mice here on the left have inherited this YFP allele from their mother. This is a confocal, or this is a immunofluorescence image of the cerebellum. And so the red here is a nuclear marker, the blue is an astrocyte marker, and the green is UB3A. And so you can see there's very high levels of expression of UB3A in these Purkinje neurons, as you would expect. However, if we look at a mouse that's inherited that YFP allele from its father, we see that it's basically been turned off. And that's what we expect to see. But what's nice about this mouse model is that it's a very nice reporter for looking at the allelic expression of UB3A. So we took mice that had the paternal YFP or a YFP allele on the paternal chromosome, and we isolated embryonic stem cells from these mice. And what we do with this is we go through a process where these embryonic stem cells are expanded in culture. And this is the work that we do with the Texas A&M Institute for Genomic Medicine. We have a system that we've set up where we start on a particular day. A vial of these embryonic stem cells is grown in a dish. Remember, they have a paternal YFP allele that's there. They will grow these until there's roughly 500 million cells. So there are many more than what we would get from one mouse. And then we're not doing this with a live mouse. We're actually doing this in a dish. And then what we do is we treat those embryonic stem cells with a certain cocktail of hormones and reagents that cause them to differentiate into neurons. And so instead of having to isolate a neuron from the mouse brain, we can create one in a dish. And since we can start with 500 million cells, we end up with a lot more material that we can use to screen our drugs with. And so this is what we've spent a tremendous amount of our time on for the past 2 years, is setting up this assay so that we can look at thousands and thousands of drugs. In addition to that, our collaborations with the Institute for Biotechnology in Houston, the high throughput screening facility there it's an automated system, so there are robots and automated imaging machines that will seed these cells into 384 well optical bottom plates. There's very little human interaction here. There's mostly robots that are doing this. The compounds are added by a robot, and then the cells themselves are actually imaged on a machine. So there's actually no person. There's a person that's running this, but it's all an automated process. And then what we do is we take this data back and we mine it to see if we can find things that are capable of reactivating expression of the paternal allele. And so much of this work has been done by Jade Benjamin in my lab. And so she spent quite a bit of time demonstrating that this assay will work. You can see here that on the left, these are embryonic stem cells that have been differentiated in neurons. And so there's the red here reflects the neurons. The cells that don't have red represent other cell types. So this is a mixed cell type that we have in there, but much of them are neurons. And then you can see here that if we know that when these neurons differentiate in culture, that UB3A becomes imprinted like it does in the central nervous system. So these embryonic stem cell derived neurons recapitulate the inactivation of the paternal allele. So she demonstrated that was the case. And then she also used topotecan as a positive control to show that she can reactivate it. So for us, this was just a lot of time with the assay development and getting this and demonstrating that it does in fact work. To fast forward through about a year's worth of work, what we've done is we've screened roughly 16,000 drugs. And again, this would be a very different experiment if we had done it the conventional way, but because we had set this assay up to do this, it actually could go well beyond this. In fact, this many drugs can actually be done in 2, perhaps 3 rounds of screening, which once it's running, it's a very powerful system. We've stopped here, so we've been mining this data to see what we found. We picked up 3 novel hits in the NIH clinical collection. So this is a series of FDA-approved drugs that were available to us, and we found 3 compounds in there that were, at least on the first pass, capable of reactivating expression of the paternal allele. The disclaimer here is that we have to go back and validate all of these. I expect that, not most, but some percentage of these are false positives, but nonetheless, this is the process. We picked up 1 hit in the Prestwick Chemical Library, and then we had 31 hits in a Maybridge library, which is a small molecule library. So these are compounds. They're not drugs, or they haven't been tested in any way, but they're thought to represent all of the small molecular space that can be made up with certain combinations of atoms and so forth. So what we're doing is we're going back through this data and validating all of these hits to see which ones we can throw out and which ones we'll pursue further. And here's just kind of an example of what this looks like. So this is one of the drugs that was picked up in the NIH clinical collection. And so this is sort of what the output looks like from the experiment. This is an entire well on a plate, and so there's about 300 of these that will image on one plate. We usually run 12 plates at a time. The blue image here, it's a dye for the nucleus of the cell, so you can tell where the cells are at. This red here represents shows where the neurons are at in the dish, and then the green is the UB3A protein. And so in a control, this would be black. So we know that the paternal allele is almost entirely off. It's a very a robust screen. When it's reactivated, the neurons fluoresce. And so this just gives you an idea of what that looks like. And then here's another one that we picked up in the small molecule library. So again, we have to go back and validate these to show that there's not false positives. Some of these compounds are actually autofluoresce. So they're just by shining light on them, they'll give these colors. And so we want to make sure that's not the case. But nonetheless, to just give you an idea of what we've done with this and what we've found so far. The next experiment that we have is our symptom-based approach. And again, this strategy here is not so much to target a particular pathway or gene or mechanism. This approach is simply to find something that mitigates the symptoms associated with Angelman syndrome. As some of y'all may or may not know, there's a fly model. There's actually a number of fly models for Angelman syndrome. And perhaps not surprisingly, those Angelman syndrome flies recapitulate many of the symptoms seen in Angelman syndrome. So for example, the flies have motor coordination defects. They have learning and memory defects. They have circadian rhythm defects. And we can use that as an outcome measure to screen potential candidates. And you say, well, why do this in a fly? Why not do this in a mouse? Well, mice are very cheap. You can grow lots of mice and have large colonies of mice, but surprisingly, they are very expensive. Their life cycle is, a couple of months. My bills, I know that all of us here, we spend thousands of dollars each month on maintaining our mice. The best labs, and from what I gather even from some of the best pharmaceutical companies, can only screen a few compounds in mouse models, just simply because of the time that's associated with it and the time that's associated with maintaining the mice and so forth. Flies, on the other hand, can literally be grown for pennies. I'm sure all of y'all had fruit flies in your house before, right? They're almost impossible to get rid of. You can have tens of thousands of these animals in a lab and maintain them with very little cost. The life cycle of a fly is about 10 days. Flies start having more flies the day they're born. So it's a very robust, very high throughput assay that you can use that has a phenotype that you're interested in. It's probably the lowest organism on the list of those that actually manifest some symptom that you could do this with. Now there's a lot of biology between a mouse, or between a fly and a mouse, and especially a fly and a human, but this does work, and there are examples of that. Investigators have used the Fragile X syndrome fly to discover new drugs. There have been cancer agents found, or cancer drugs found in fly models. It's very different, and I can understand what it must be like to hear this, but it is a very powerful and very quick way to screen things. And actually what you can do here is you're in the order of thousands of compounds that you can look at. It's not as high throughput as, say, a cell-based assay, but it is very high throughput compared to other organisms. And so I'll just kind of give you an idea of what we're doing here. This is something that we've been setting up the assay for this. We're using 3 different approaches, different growth chamber formats, if you will. So we can grow these flies in 12 well vials or plates, 24 or 96, and I think there's 24 there or 12. And then these different 96 well formats. And so each one of these tiny wells can be home to a dozen or so flies. And then in each one of those wells, we can add a particular drug to it. So on these 96 well formats, we can screen almost 100 drugs at one time. And that's pretty amazing when you think about what it takes to do that in a mouse. What we're focusing on right now is larval locomotion, adult locomotion, and circadian rhythm. And again, we're simply looking for things that rescue or mitigate these symptoms, with the idea that it's something that's found will then be validated in a mouse model and then perhaps in another animal model, which leads me to my next topic. So it was actually one of these meetings a few years ago that this idea about generating a large animal model of Angelman syndrome really hit home. And so just a little brief background, my Ph.D. training was in large animal genetics, and my Ph.D. advisor there at Texas A&M was a pioneer in developing transgenic animals, particularly pigs. And the idea was that the pig model would be used to replace the mouse model. And there are a lot of reasons for wanting to have a large animal model for a condition, and being involved with this group really proved that to me that was the case. And the idea behind this is that for those compounds that are found, whether it's through the cell-based assays, the fly assays, the work that Dr. Weber or the others are doing, that has typically ended in a mouse. And that's usually been the gold standard. For preclinical studies, most groups will look at the efficacy in a mouse model, and then they'll test the toxicity and the pharmacokinetics in a large animal model, for example, a beagle or a dog or another breed of dog or a pig. Never has anybody had a large animal model of a condition. Now, they do exist, and they happen naturally. And typically you'll see veterinarians stumble onto them, and they identify them and they maintain them. But only recently have people been able to design and generate these large animal models. And so what I mean by that is actually creating an animal that has a mutation in a specific gene. And so what we proposed here was to make a pig model of Angelman syndrome. And so as I mentioned, my background or my training started in this area. My PhD advisor, Dr. Jorge Piedrahita, has left A&M and has been at NC State University for the past 10 or 12 years. He's the director of the Center for Comparative Medicine and Translational Research. It's actually, I believe it's an institute now. And there's a series of events that have led to this, but there's only a few groups in the world that can actually do this, and he happens to be one of them and one of the best of them. This particular project that we have involves a large number of groups. So we're working with NC State to develop the pigs. We're working with veterinarians at Texas A&M who will help us with characterize the model. Dr. Gwen Levine is an expert in gait analysis in animals, and we actually at A&M have a gait analysis system very similar to the one that Dr. Weber showed, that is on par with what you'll see in any hospital in the United States. And it's used entirely for large animals. And so we're working with groups that will help us characterize these pigs once they're made. Also, Dr. Ann Anderson and Dr. Weber will be helping us with the EEGs in these pigs and the learning and memory of these pigs. And then also we'll be collaborating with Dr. Siegel in terms of the design and some of the therapeutics that we're going to test in them. And the idea behind it is fairly straightforward. So as I mentioned, preclinical studies involving new drugs jump from a mouse to a human. And unfortunately, there are examples of many drugs that have failed after this jump. And that can be explained for lots of reasons, but one of them can be that there's just too much biology between a mouse and a person. And so the idea here is to put something in between the two. And so a pig is, physiologically, it's much more similar to a human. It's a large animal model that's been used for decades in biomedical research. The pigs were used extensively for heart transplants and stents. As I mentioned, they've been used to test pharmacokinetics and toxicity of drugs. And so there's a lot known about pigs in terms of their physiology, their anatomy, and so forth. They are a litter-bearing animal. They have a gestation of 3 months, 3 weeks, and 3 days. They're relatively inexpensive to maintain compared to companion animals or to non-human primates. And there aren't the ethical or societal concerns that are associated with using them as a model system. And so we felt like the pig, having our resources at A&M, my interactions with Dr. Ph.D. and my background in this area, to develop a large animal model of Angelman syndrome that would be used solely, if not exclusively, for testing those most promising candidates before they're tested or used in humans. And you can imagine how this would unfold. If there's something that's promising that's found in a mouse, and it's tested in a pig, and it's also shown to be effective in terms of its treatment, well, then your confidence that it's going to work in a patient is heightened, elevated. If there's a promising treatment found in a mouse and it doesn't work in the pig, well, now you have the opportunity to decide whether or not you want to pursue that. I don't think anybody wants to be testing these things without some degree of success associated with the treatment. And what this, or at least the idea behind this, is that it will ensure what we're using will be effective. It'll also give us a place to work out the dosage. The pigs, commercial pigs that become adults are very large, but there are strains of pigs that, the miniature strains, that as adults are approximately the same weight as humans. And so there's a lot of actually research going on using these as models, and we foresee that this will be the case for us too as well. And so I've sort of mentioned this here. The physiology is very similar. The metabolism is very similar. Brain development is very similar between the pig and the human. And perhaps not surprisingly, cognition. Pigs are all actually thought to be smarter than dogs. They have a very complex social hierarchy that exists, and so there are a number of things that we feel that we can use or test in the pig that we wouldn't be able to do in any other model system. It's been used extensively for other diseases. As far as, to the best of my knowledge, this will be the first project where it's a pig's being used to study a neurodevelopmental disorder, but nevertheless, this has been used and been very successful for other conditions. Based off where our location at Texas A&M University, we have all the resources and infrastructure to characterize these pigs. A&M is also the home of the Texas A&M Institute for Preclinical Sciences, or Preclinical Studies. It's actually a GLP facility that was designed to test pig models for various uses in biomedical research. This is all the work that's being done at NC State. This has to be validated, but there is evidence that we've deleted the UB3A gene, the pig UB3A gene, and now what we'll do is proceed with making the pigs and then characterize them. So hopefully by next year we'll have a lot more to show you in terms of what these animals are like and what we're doing with them. And then with that said, our design, as you can see here, is to use a high throughput assays both in cells and flies. To collaborate with those groups that are using the mouse, we also have mouse models of AS for the most promising candidates, and then the top candidates would then be tested in the pig model, and then those that pass that would then go on for use potentially as a therapy in the patients. And with that, I'd like to thank everybody in my lab, most of them that I've already mentioned, as well as the fire team and FAST. Thank you.